Okay, so I've been listening and thinking to a lot of what we've been chatting about in terms of notes. So what I'm going to do is anything that's really on the white A4 paper can be converted into notes, and that will give you the bedrock of the information that we will cover. I do feel strongly that although notes are good, it is only part of the system of learning that we're going to need to try to get you to really find the best way of understanding all of this as you can. So I'm not going to give up on some of the other things, but I'm going to put them specifically at the end of the video. And if you're just wanting the notes at this stage, that is totally fine. And you can skip out the last bit. I will be doing some work with that PyMol program that we used a few lessons back and showing you really how DNA wraps around histone proteins. It's quite a cool little bit, and it's interesting. However, we shall crack on. This topic, well, this section is really about ligands. Now, ligand is actually a word that has two meanings in science as far as I'm aware of it. It's probably got even more. Specifically in biochemistry is any molecule, atom or ion that binds to a protein. Now that is very different from what a ligand is in other types of chemistry, and you might see that word in a different situation, and interestingly the meaning of what it's trying to get across is almost flipped on its head. So be careful with what people are actually trying to say with the word ligand. The only thing that you'll come across with ligand in advanced higher chemistry is the biochemistry meaning, and that is that any molecule, atom or ion that binds to a protein is a ligand. However, if you're doing advanced higher chemistry, you will come across this word and it will mean a very different thing, almost the opposite thing, where I'm not even going to confuse us by going into it, but basically the meanings are almost reversed in advanced higher chemistry. So just be careful with this word. We are only going to be speaking about it in terms of biochemistry, and that is really your go-to definition for ligand. Right. The most important ligand, I think most people would probably not argue, is DNA. So DNA will interact with a very specific group of proteins called histone proteins, and they will form the structure chromatin, which will help really give you your chromosomes. That chromatin really is a chromosome in short. So the chromosomes that contain genetic information are a combination of the DNA, which is in orange here, and the histones, which I've done in that light blue colour, aqua blue, maybe aqua green, whatever you want to call it. And that DNA wraps around the histone proteins. Any single one, and at the very end when we move on to that PyMol program, I'm going to show you a single one. So one histone protein plus the DNA that's wrapped around it is a nucleosome. It's worth saying that that singular histone protein is a good example of a quaternary structure. They are pretty bulky proteins, and they are made up of quite a few different subunits all put together. Now, the combination of that histone protein and the DNA, if you're just looking at one, is a singular nucleosome. If you're looking at lots and lots of those all together, you're really saying that it's chromatin. And if you're taking a view of the whole structure of that chromatin from start to finish, you're looking at a chromosome. So the nomenclature here is quite tricky. It's taken me a couple of years to really wrap my head around it properly and know how to talk about it and when to talk about it. I would say it's probably one of the hardest things in the biochemistry / genetics is the wording, because the wording has evolved over time, and it really is like learning a little bit of either a new language or a new branch of English. So it's worth getting your head around the wording, and being clear with yourself about what you mean when you say what you say. That will really help you build a picture of that. So that, to me, is arguably DNA is the most important ligand that you could think of. However, it's a pretty odd ligand. The reason it's quite an odd ligand is it's much, much bigger than the individual proteins themselves, and it's arguably the more important of the two. If you're going to have that debate, what's more important, the histone or the DNA, you could probably argue the DNA itself is more important. That's going to be flipped in in the examples that we come to later on. But for this one, it's an interesting example, and it is a little bit different. Most ligands are you'll probably find will be smaller than the protein itself, and the protein out of that partnership would be considered the more important of the two. But this is our first example, and it's a little bit of an odd example, but it's an interesting one. And to think of DNA in this context is quite interesting. Right, so to go over why DNA and the histone proteins interact in the way that we do, we've got to take a little bit of a look at the DNA structure itself. Now, the double helix and the way that the topology, the way that the DNA contorts itself, can actually change. It's a really interesting branch of study, and there's actually different names for different ways the DNA can really wrap itself up and how its shape forms. But the important thing for this is that we, for a very long time, have focused on the bases holding the two strands together with their hydrogen bonds. However, in the case of their interaction with the histone proteins, what we're really looking at is that negatively charged backbone. The reason that it's got quite a strong negative charge is that it's a sugar phosphate backbone, and there is a ring of sugar with a phosphate, and there's a pretty heavy negative charge that comes along with that phosphate. That negative charge will be attracted to the positively charged parts of the histone protein. That's really what I want to highlight later on as well. We're going to take a pretty in-depth look at that histone DNA interaction. So the histones have positively charged sections that DNA will interact with, and it will hold it wrapped around. In fact, it will wrap around quite a number of times any given histone protein. So that negative charge of the DNA backbone is attracted to the positive charge of the histone protein. Right, what we're going to do is we're going to move on from DNA, and we're going to take a look at more classic examples. We're going to retread some areas that you will have hit in higher, and we'll discuss it a little bit, but we will, for the time being, we'll leave that DNA as a ligand almost in the past, and we'll talk about some more classic ligands. So our more classic ligands are usually a little bit smaller than the protein itself. So what we've got here is that our substrate, really going back to higher, will have an impact on the shape of the the enzyme that it's interacting with. So the shape of the active site will be forced to change by electrostatic interactions with the substrate, and that shape change will allow for greater binding. It will increase the affinity of the active site for the substrate. Now, it isn't just the substrate that can have an impact on the conformation of that active site. You may have a ligand that can bind onto your protein, and that could affect the shape of the active site as well. So in this case, this ligand, and we'll come on to this a little bit later, might be a negative modulator, so it would change the active site in a way that would inhibit the enzyme. This is really important for controlling enzymes, because once you've made an enzyme, you may want it to only activate under certain conditions. Certainly for pepsin, you're only really wanting that to break down proteins in a very specific environment. You're not wanting that to be ravenously chewing through your body's proteins. That would not be good. So it's really important that you maintain control of enzymes, and you can do that through these smaller ligands. So once again, this is more a recap of induced fit, and it's the idea that the active site without the substrate doesn't actually have the right conformation to fit the substrate, but as the substrate approaches that active site, there are changes to the shape of that active site, increasing the affinity and allowing the substrate to bind. Once the substrate has bound, and maybe make this a degradation reaction, it breaks up. The products have a low affinity, so low affinity products and high affinity substrate, and it's those interactions that will help change the shape of that active site. So when we're talking about electrostatic interactions, really we're talking about those hydrogen bonds, dipole to dipole interactions, and to some extent London dispersion forces. But as we covered before, those are really pretty weak, and I'm struggling to envision how big an impact that would have on an active site itself. So really hydrogen bonding and dipole to dipole interactions, remembering that dipole to dipole interactions are really just weak hydrogen bonds. Right, so those ligands that we were talking about that can interact with the enzyme can really do one of two things, and to be honest, this is probably one of the more important notes that we've got today, and the wording is going to be quite important. I can visualize the SQA creating questions about this already, so this is a pretty important little bit. So altered activation energy, really you can change the affinity of an enzyme for its substrate with ligands. So you can chuck something into the mix that's either going to make your enzyme work better and more effectively, or cut its activity down so that it isn't able to do its job as effectively, and that might be really important. Basically, we've got an accelerator and a brake, and it's just the names that you've got to learn for that accelerator and brake. So ligands can bind to the allosteric site. That is the site, any site on the enzyme that isn't the active site that will bind to the substrate. That ligand binding to that allosteric site will cause a conformation change or a shape change, same thing, in the enzyme, or to the enzyme, however you want to say that. Now, that ligand binding to that allosteric site will branch into two potential outcomes. Either it will increase the enzyme affinity, or it will decrease the enzyme affinity. So aka it will make it easier for the substrate to bind to the active site, or it will make it harder for the substrate to bind to the active site. Now, ligands that do this, if it's an increase in enzyme affinity, that is a positive modulator. That is your accelerator, so it will make the enzyme work more effectively. If you're decreasing the enzyme affinity, that is a negative modulator, and that is your brake. You're going to slow down that enzyme, and you're basically going to stop it functioning as well. So that is probably one of the more important things. We will, as time goes on, talk about some specific examples of positive and negative modulation. That is probably something that is worth a little bit more of a discussion going forward. Right, we're about to move on to a bit that changes tack a little bit, and although ligands are going to be spoken about in some way, I'll try and highlight when that is. The next bit is, to me, something that kind of came out of the blue in terms of the notes. It's quite abstract in terms of what I would regard as, like, this is all par for the course, but the next bit really is quite interesting. It's interesting that it's in the course as well, and it's certainly something that's worth paying a little bit of attention to and having a little think about it. It was new to me. I had never come across this before. So we'll go on to quaternary cooperation and cooperativity as the next bit. So this is our quaternary proteins having the individual polypeptide subunits having an impact on the conformation of the other subunits that are there, and we'll talk a little bit about this. So when there is a change to one polypeptide in a quaternary protein, this may have an impact on the conformation of the other subunits. If it does, this is called cooperativity. So we've got whatever this whole thing is our quaternary protein, and it has 4 polypeptide subunits. So this protein is created from 4 polypeptides all interacting with each other. Now, if a ligand was to bind to 1, then 2, 3, and 4 would be changed in such a way as their activity would increase. Now, the example for this that is spoken about is hemoglobin. So there is 4 heme groups in hemoglobin, if I'm right, I think. It doesn't matter really about the numbers. It's more the idea that's important. So if there's 4 heme groups, there's 4 positions for O2 oxygen to bind to, and they will be binding to the iron ion. Now, as soon as you get oxygen binding to 1, the affinity for oxygen in the other subunits increases. So as soon as oxygen binds, the affinity that the rest of the protein has for oxygen goes up. AKA, if there's 1 oxygen bound, it's more likely that the other subunits will have oxygen bind to it faster. Why this would be important, or if it's in terms of evolution, or whether or not it's simply a chance of chemistry, I'm not actually sure. I would need to take a look at that. However, It's certainly something that's in the course notes. It's not something I know a huge amount about, but really that's all we're saying. We're saying as soon as a ligand binds, the affinity for that ligand in the other subunits increases, and so if you've got one oxygen bound, it's going to be easier for oxygen to bind to the other sites. Right, we are actually almost through this section of the lesson, so I'm gonna keep going with probably our most classic ligand, and this is going to be very different. Well, I guess it's not all that different from the DNA. We are talking about a phosphate group binding to a protein, so that's very interesting that this is a real classic one, phosphorylation. And although I was about to say it's very different from the DNA binding, it's actually a negatively charged ligand binding to part of a protein. This is used in a very different way, however. So this is really a very important post-translational modification, aka we're changing it after it's already been made. So phosphorylation is the addition of a phosphate group to an R group of an amino acid in a protein. Now, once again, you might see a phosphate group denoted like this or like this. Really, the more acidic the environment, the more likely it is to be this. The less acidic the environment, the more likely it is to be this. This gives you probably a better image to have in your head because it's got the negative charges. Especially if you're not into your chemistry, you can forget that those hydrogens can pop off, leaving a negative charge. So it's probably best to have this one in your head for a phosphate group. So this is you're going to be bored of phosphorylation before the end of Unit 1, but it is a major trigger for protein action. So it's a way of making a protein do a function, and we're going to take a very specific look at that in a minute. So the enzyme kinase is really responsible for the addition of the phosphate group. Now, you don't need to know this bit actually. Technically, this bit is a little bit over and above, but it's interesting to know. So the targets for this are usually serine, threonine, and tyrosine. Those are the common amino acids within the protein that will be targeted for this phosphorylation. Once that, you can kind of imagine it like the addition of the phosphate group, and then this bit of the protein will change shape, or it will carry on an action, and it's very important, especially because our main route of transforming energy from sugar is to make ATP, which is adenosine triphosphate. So this phosphate ion is really important in biochemistry, and it's something that really your body uses all the time to take action. Right, so the very last little bit of this before we go on to the pymals. Okay, so one of the classic uses of the phosphate ion is in ATP, and what we've got here is the myosin protein with the myosin heads that basically bind to and drag actin towards the midline. So what's going to happen during a muscle contraction is an ATPase enzyme is going to add ATP to the myosin head. Once that happens, the myosin head attaches to the actin filament, and in losing the phosphate So one of the phosphates from the ATP to make ADP plus inorganic phosphate, we drag the actin towards the center line. That action contracts the entire long cell of the muscle fiber, and that is basically how your muscles move, that contraction action. And that is the use of a phosphate ion which has already been bound to ATP. So that is muscle contraction, and you are using the myosin heads and the actin filaments, and the myosin heads will bind to the actin and drag them towards the midline. We're actually going to take a look at an experiment that has this, that can show this even with tissue that's not in a living organism. So muscles will, the myosin and actin proteins will still do this action even when the rest of the animal is dead if you provide ATP. So that is actin and myosin. ATPase is used to add the ATP to the protein, and then that breaks down, giving the protein energy to contract the actin fibers towards the midline.
Okay, so this is us sitting with our nucleosome, and you can see the DNA is wound round this really large green, for the main part, central structure. The little dots that you can see, the spheres around the DNA, are manganese ions. You wouldn't see these, I don't think, in vivo. I believe they should be magnesium ions in vivo. But essentially those ions are there to help stabilize the DNA structure. Now, all the bits of the DNA that are in that kind of goldy orange colour, that is pretty strong negative charge, and throughout this whole central structure we've got lots and lots of alpha helixes that make up our histone protein. Now, it looks pretty complicated and pretty horrendous at the moment. What I've done is I've pre-selected a few things to take a look at. We can discuss them, and that will be kind of the overview of your nucleosome. So if we go to, I've selected the ones to hide. I want to hide that. I want to hide that. Really, I've just hidden those alpha helixes of the nucleosome, but I have left a little bit of the histone protein. I've just selected this little bit here so we can see the importance of the charges, really, and where those charges lie in relation to the DNA. So what I've done is I've got my positive charge here. I'm actually going to change the colour. I'm going to make it a bright green for the positive charges, and the negative charges I will make a... Ooh, what will I make it? Magenta. That sticks out not too badly. Well, guess we could make it orange. Yellow worked fine. Make it yellow. So, the negative charges are from our R groups with a big strong negative charge, and our positive charges that are coloured pink, they are from our R groups with a nice old positive charge. Now, if we take a look at where the DNA is, hopefully do is try and zoom in a little bit on where we are. So that positive charge on that yellow, it's a little bit tough to see, will be interacting with the negative on the DNA. Now, where you've got a negative charge on the histone protein, that's actually not going to be orientated towards the DNA. It's going to be orientated away from the DNA. And you will find that there are some negative charges on the histone protein, but overall it's much more positive. So the areas of yellow are certainly more numerous than the areas in pink, and that is a really good example there. So this strand of DNA here, all negatively charged, is interacting with all of this curly bit of the alpha helix, which is positively charged, and that's holding that strand in that position. So that's actually a nice little just here. That's a nice little show where that interaction's happening, and we've got our positive charge here and our negatively charged DNA backbone. It is something to get your heads around, is the structure of chromatin and how important that is, and to build up a picture of it in your head. There's lots and lots of work that goes into what DNA looks like when it is interacting with histone proteins, what DNA looks like when it's interacting with these other ions. So just to reiterate, and I'll go back to showing the rest of our histone protein. The DNA in this case is a ligand. So I wonder if I can center it. The DNA is really a very, very large ligand, and as we were chatting about earlier, it's arguably more important than the actual histone protein itself. This is histone H4, I believe. So there are different histone proteins, but most of them do a similar job. Right, hopefully that stuff was pretty clear, and you've had a little look at our nucleosome. A bunch of nucleosomes all together will be chromatin. Chromatin will form into chromosomes, and it's your chromosomes that contain your genetic information. So that is our stuff about ligands, and I am gonna get our next lesson up by Friday. I'm running about a day late this week for numerous reasons. But we should catch up if I can get one out by Friday. I'm also going to try with Dr. Hayhurst and run a Q&A so that we can see if we can finally get those Q&A things on Teams running. Right, guys, you have a good day. Just notes for this one again. We're almost to the point where we're going to need to recap and go over things, but we've got one in bits and pieces to do before we get there, but yep, that's it. Right, goodbye.